Welcome everyone to a brief introduction to Dbrain. So all of those you see here in the Zoom window are part of Dbrain and contributing actively. And in addition, we have additional people on board that could make it today. So I'm Jeanette Hellgren-Kotaleski, professor at KTH, and I will start with a brief introduction, and then we will see some highlights on ongoing science. So let me share the screen. So Dbrain stands for Digitizing Brain Data for Health and Disease. And in neuroscience today, we have a lot of data. We can measure things over various temporal and spatial scales. So we have a lot of data, and we really, if we want to go from data to knowledge and understanding, we have to capitalize on the possibilities to digitize all this data. So Dbrain is a collaborative project within the Digital Futures framework, and it also relies on multidisciplinary collaborations between mathematicians, computational scientists, health scientists, and we also have experimentalists from the Karolinska Institute on board, as well as clinicians from the Karolinska Hospital. So why Dbrain? Well, first of all, a third of us will unfortunately suffer from a brain disease over our lifetime. And this creates a lot of suffering also for our relatives and families and friends and so on, and it costs a lot. More than a third of the healthcare cost in the Western world is due to brain-related disorders. So today, when a patient feels ill, he or she goes to the doctor, and the doctor probably takes some tests. We will call that biomarkers. And then based on these biomarkers, the doctor will set a diagnosis and start a treatment plan. And this is of course fine, but we can do better than that if we use data analysis methods and apply it on all these biomarkers. Then we can both help the individual patients and maybe better give a treatment plan, but we can also help different groups of patients better. And to finally nail down more of the underlying mechanism to try to understand this causal chain of events, we also need to apply modeling and simulation. And this is what Dbrain is about: data analysis and modeling and simulations. So we focus on a couple of challenging questions. One thing is, what is the relationship between the brain structural connectivity and functional interaction between different brain regions and within the local networks? So this is one of the holy grails within neuroscience, like structure, function, dynamics. Can we predict one based on the others and so on? The other big challenge is, how do slow changes in the brain's chemical balance and the gene expression patterns and morphology changes shape the dynamics function of the brain? Because over our lifetime, we have these slow changes, and also when something goes wrong and we get a brain disorder, also these changes occur, different changes occur, and we want to know what is actually compensatory mechanism or what is causing the disease. So we will focus on Alzheimer and Parkinson's disease because there is a lot of data on that, and also those are the two most prevalent neurodegenerative disorders. So this is how Dbrain is organized when it comes to data analysis and AI-based data analysis. We apply topological data analysis and also machine learning approaches onto these biomarkers data, including brain imaging data. And when it comes to modeling and simulation, we model over multiple scales. And then the goal is to enhance and improve the diagnosis and prognosis. So these are the original PIs. And now, of course, also many postdocs and students are involved. And you will hear now a few highlights of ongoing work from machine learning applied to brain imaging data, from topological data analysis field, and also examples of how modeling and simulation might help. now over to next presenter.
Yes, so my name is Rodrigo Moreno, and with Orion Smetby, we are working in medical image analysis in Dbrain. So these methods are used to extract information that can be used for different purposes. So, for example, you can use images for diagnosis. So the doctor can ask for an MRI and see if the hippocampus is shrinking. So then this is a sign of dementia. Or it can be used, for example, in this image of the coronary arteries, that this person has some stenosis, but these images can be used to select the best treatment for this person. Or it can be used also for assessing the effect of a treatment, just taking images after the treatment. So then the doctor can just use that information to change the treatment if it's not working, and so on. And there are many other applications that can be used in clinical practice for these methods. The traditional image analysis methods are works like this. So assuming this is just an example, we want to discriminate MR images between two groups, patients and healthy. And then the standard methodology is to use handcrafted features with rules given by experts. So I was mentioning before the shrinking of the hippocampus. So then this is knowledge given by the doctors. So the idea of conventional machine learning is to just let the computer learn these rules. So not the doctor anymore, but just give some data. So then the computer can learn these rules to distinguish between patients and healthy subjects. And deep learning, that is the current status of artificial intelligence, is an end-to-end solution where the data not only discovers these rules, but also to extract the most relevant features. So then the involvement of the human is quite reduced. In Dbrain, we are working with connectomics, which is the study of the brain connectivity. And there are two types. Structural connectivity looks for the physical connections of different regions in the gray matter. And this is done with tractography on diffusion MRI. But you have also functional MRI, functional connectivity that comes from correlation of the activity patterns in the brain. And this is, we use resting state fMRI for that. And we are doing AI methods for connectomics in our group. So how it works for structural connectivity is that you have the diffusion MRI data, you compute tractography, so these lines that you have here, and then you can compute a brain connectivity matrix. And we have proposed methods for improving the quality of this tractography that is always challenging. So for example, here in this method, we developed an AI-based tractography filtering. So the idea is to generate with tractography a tractogram with a lot of streamlines, and then just to extract the ones that are relevant for that are plausible. And the problem is that we don't have a ground truth available. So then we have to use some kind of supervisors to estimate the plausibility of these streamlines. And we found out that about half of the streamlines are inconclusive. So we don't really know if they are good or not. So there are a lot of work that we are going to tackle in Dbrain for reducing this number of streamlines. And also we, in another project with a student, we tested the effect of filtering tractograms for classification of patients with dementia. So we use these brain connectivity matrices to see if we could distinguish between patients and controls. And with and without using filtering. And we found out that filtering improves the classification of patients by 5%, which is a lot, I think. In another project, we used another method for tractogram filtering that is called SIFT. It's a very advanced method, but we couldn't really use it in our previous work because it mixes non-plausible and redundant streamlines. So these redundant streamlines are good streamlines, but they are just discarded by the method because they are redundant. And we don't. This is a problem for AI because we need to distinguish between non-plausible, redundant, and the correct ones. And we did something that is called randomized SIFT. So we use AI to improve this method, and then we got very good results of 80% accuracy for separating non-plausible and redundant. And we can see here the blue dots here are streamlines that are positives, the yellow ones are negatives, and the black ones are the redundant ones that are very close to the good ones, the plausible ones. Also, we have been working with segmentation for Alzheimer's disease. So in this work, we wanted to segment the hippocampus. As I said, that this is very relevant for Alzheimer's disease. And what we did was to use deep learning twice. In the first step, we estimated the shape of the hippocampus, and in the second step, we use another deep learning network that considers this shape of the hippocampus, and the result is that we improve generalizability, so that if we use this method in unseen data, it performs better than previous methods. So that's the current status of our work. So I will leave the word to Martina.
I will share screen. Hello, I'm Martina Scolamiero, and together with Wojtek Hachulski, we work in topological data analysis at Dbrain. For us, topological data analysis is about vectorizing geometry. In general, data analysis is about simplifying, forgetting irrelevant information that floods and obstructs our understanding. In topological data analysis, we extract features invariant signatures based on geometry, which are suitable for statistics and machine learning. These methods are abstract and can be applied to different types of data, ranging from brain reconstructions at the level of neuron and synapses. Here on the left, you can find a striatum reconstruction representation by our Dbrain collaborators, Johannes Jorf and Alex Kozlov, to galaxies in the universe, represented here on the right. But what does based on geometry mean? Classically, geometrical techniques are already in data analysis. For example, approximating bilinear subspaces includes principal component analysis and regression. Kernel methods are about scalar products and angles. Connected components are found by clustering methods. These classical methods are just example of a comprehensive framework and approach to geometrical data analysis that TDA offers. Based on geometry means that the first step of retrieving information is to transform the data into spatial information. Schematic representation here is on the top of a geometrical object. The aim of this step is not to simplify, but rather to enhance complexity through geometry. This complexity might be compressed in its original form, and the decompression process allows sometimes to see patterns and regularities relevant to our problem that the compressed original form of the data was hiding. Having spatial information, we can then simplify what we see into various geometrical summaries. The simplest one are, for example, counts of various motifs, clicks or simplices in the geometrical representation on the data. On the right, you can see simple type of motifs, which has been really useful in a recent research to neuroscience. And we can here see representations of directed simplices of dimension 2, 3, 4, 5, 6, and 7. These are structures in the data, cliques of all-to-all connected nodes, which have an inherent orientation from a source to a target, and therefore inherit a flow within the dataset. Here is an illustration of how those click counts distinguish microcircuit reconstructions of striatum at different stages of Parkinson progression. This is an ongoing project with Kotaleski's group within Dbrain. Another way of extracting simplifying invariants out of spatially decompressed data involves homology. Here is an illustration of how homology-based simplification developed by our TDA group at KTH, called the stable rank, can be used to distinguish basic geometrical features: circle, rectangle, triangle, and an M-shaped segment. I will now leave the words to Arvin. Thank you.
My name is Arvind Kumar, and I will present you the third aspect of Dbrain. So first two aspects you heard were about measuring the structure and quantifying the structure. But brain diseases, they are not just manifested as changes in the structure. The structure leads to change in the activity dynamics of the brain, and that manifests as the diseases. And we think that we could use our understanding of the dynamics of brain networks to comment more about brain diseases, create new kind of diagnosis, and hopefully ways to control the brain dynamics, which could then eventually become maybe a treatment for the diseases. We have hired 3 postdocs for this purpose, Henry, Pascal, and Boo. They are in this session, and these are the 3 people who will be working on this, exploring how brain dynamics is related to brain diseases and how structure leads to changes in the dynamics. I'm going to give you 2 quick examples how we can use brain dynamics or understanding of brain dynamics to think about brain diseases. The first study is basically focused at finding out what exactly changes as the brain degenerates. We are focusing on Parkinson's and Alzheimer's disease. In both diseases, there is neurodegeneration happens. And question is that, what does it lead to? So to study this question, we investigated 3 different models of brain connectivity. One is random network, another one is small world, and the third one is scale-free. And in these 3 models, we removed neurons as well as synapses, mimicking some sort of disease progression. And as a consequence, we measured what changes in the activity of these networks. And it was somewhat a bit of surprise that in all these 3 networks, if we start removing neurons or synapses, synchrony, which is shown on the x-axis, and firing rate, which is on the y-axis, they both change in a systematic way, irrespective of how we degenerate the network and how we, what kind of network we start with. Also somewhat surprising is that the consequence of change in both neuron and synapses, that's same. And when we investigated which part of now the network structure relates to the network dynamics, we found that what really matters is the effective synaptic weight, which essentially refers to balance of excitation and inhibition. That is a predictor of these different dynamical states, be it the firing rate or synchrony or regularity of spikes. So this tells us that irrespective of change in the connectivity, one of the main parameters of the network that changes is EI balance. And this is something that we will be focusing in future, how we can infer it from measurements of brain activity using non-invasive methods. A second example how we can use understanding of brain dynamics is in maybe classifying patients from control groups, or to find out what exactly may have changed in the connectivity of the brain at a large scale. So here what we do is we get data, functional connectivity data from healthy control subjects and patients with mild cognitive impairment. These are connectivity matrices, how different parts of the brain are connected to each other. And the question is now to determine whether there is a significant change in the connectivity or not. So we take insights or inspiration from a very common trick that we often use in our real life. When we want to know if something is broken, we just try to shake it and see if the noise is somehow familiar or unfamiliar. And if the noise is unfamiliar, we say that there must be something wrong with the system. So this is something we are calling as shake the brain. So we are mathematically shaking the brain, if you wish. We take these networks, we stimulate them with different kind of noisy and oscillatory inputs and simulate their dynamics. Once the dynamics is there, we characterize these changes in the network dynamics, here schematically shown for healthy and patients. And by comparing these dynamics of the simulated networks, we can then separate patients from control groups. And here is a nice example of this. So we measured entropy of the activity. It somehow captures how noisy the activity of simulated network is. And it turns out that patients, they have on average low entropy compared to the control subjects. And here shown for two different versions of the networks, the distribution of their activity, and here you can see individual patients. So it turns out that indeed, patients with mild cognitive impairment, they seem to have lower entropy on average than control subjects. This is of course not perfect, and we need to work more on it to explore how we can extend this analysis to develop into possibly a diagnostics. So these are two examples how we can use understanding of brain dynamics to comment about brain diseases. And this work will continue, of course, in future, and we hope that this, at least some part of it, translates into neurology, but in general, if it gives us some insights about the brain function, that's already great. So finally, this is the wrap up then. So we talked about the structure, measurement of the structure, quantification of the structure, and dynamical consequences of the structure. And these are the people who are involved in this. Janet already introduced part of this team, and this team is growing. So yeah, thank you so much.